The last little bit that we're going to talk about is genetic transfer and recombination. So how do all of these bacteria spread their genes and basically cheat? So genetic recombination is the exchange of genes between two DNA molecules that creates genetic diversity. Remember, binary fission doesn't allow for sexual reproduction. So this genetic recombination is really the only way this is possible. Crossing over has two chromosomes that break, rejoin, resulting from the insertion. So here we have DNA from one cell aligns with DNA from the recipient. Okay. Notice that there's a nick in the donor DNA. DNA from the donor aligns with the complementary bases from the chromosome. This can involve thousands of base pairs. A Rec-A protein catalyzes the joining two strands. And now we have a new gene in both. So you have crossing over or genetic recombination. Now, genetic transfer can occur vertically, transfer genes from an organism to its offspring, or horizontally, gene transfer between genes in the same generation. Transformation, genes transferred from one bacterium to another as naked DNA. So here we have a recombination event from Griffin. So encapsulated organisms were injected into a mouse. The mouse died. Colonies from the encapsulated bacteria were isolated from the dead mouse. Okay. Living non-encapsulated bacteria were injected into the mouse. Mouth remained healthy. Few colonies of the non-encapsulated bacteria were isolated. So we took heat-killed encapsulated microorganisms. So their DNA is still active. And we injected it into the mouse. Living non-encapsulated and heat-killed encapsulated bacteria were then injected into the mouse, and the mouse died. So what happened were the living non-encapsulated bacteria picked up the capsule gene from the dead bacteria and said, oh, this looks like a good fit, and then reproduced and made encapsulated bacteria that prevented phagocytosis and ended up killing the mouse. So the mechanism here, so you have chromosomal DNA, and this additional DNA can just be picked up from the environment, from dead organisms or plasmids or things that were dropped during replication or death. Conjugation is basically plasmids transferred from one bacterium to another, requiring cell-to-cell contact via a sex pilus. So here you have an F+ cell. That means it has the sex pilus plasmid with whatever genes it has on it, and it shoots the pilus all the way to the F- cell, which means it doesn't have it. Well, a unraveled piece of DNA is going to travel along that pilus and then go inside this cell here. And then that will turn that from F negative to F positive. Now, if this F negative cell had been an F positive cell, so, and again, remember, these bacteria don't have eyes really, so they can't tell, and the only way they can know is by harpooning them with sex pili. So what ends up happening is the F positive will shoot the sex pilus, and then upon contact, they'll be able to pick up if this is F positive. So if this was an F positive to F positive, this little sex pilus would just dissolve and no transformation or transmission of DNA would occur. Conjugation in bacteria. Donor cells carry the plasmid factor and are called F+ cells. HFR cells contain an F factor on the chromosome itself. So F+ is the plasmid. HFR is the actual gene on the genome itself. Remember, plasmids can be lost, whereas the ones that have been inserted into the chromosome of the cell, not so much. So this is just going through that process. This is the recombination factor, and this is conjugation. So you need to be able to describe each one of these processes by which bacteria can reproduce. Conjugation can be used to map the location of genes on a chromosome. So for example, Each one of these is coding a different gene, and this number is letting us know kilobase pairs, so how many base pairs in is this gene. Transduction in bacteria. DNA is transferred from a donor cell to its recipient cell via a bacteriophage or a virus. Generalized transduction, random bacterial DNA is packaged inside a phage and transferred to its recipient. Whereas specialized transduction, specific bacteria genes are packaged inside the phage and transferred to the cell. So this is the transduction by phage. We are going to spend more time with this when we get to chapter 13, which is the bacteria chapter. Plasmids, again, are self-replicating circular pieces of DNA, about 1 to 5% of the size of a bacterial chromosome, often code for proteins that enhance for pathogenicity and antibiotic resistance. So this is one plasmid, and here you can see, we've got some mercury resistance, some sulfonamide resistance, streptomycin, chloramphenicol, tetracyclines. This is the origin of transfer. This is the sex pilus that allows it to transfer. Conjugative plasmids carry genes for sex pili. Dissimilation plasmids encode for enzymes for catabolism and unusual compounds, such as the example of the organisms that were capable of breaking down crude oil. So they were dissimilation plasmids because they were breaking down those factors. And the resistance encode for antibiotic resistance mostly. Transposons are segments of DNA that can move from one region to another. Now, in bacteria, it's not so much of a problem. But if we have transposons in eukaryotic cells, they can be put in the wrong place and cause cancer. They contain what we call insertion sequences that code for transposases that cuts and reseals the DNA. Complex transposons carry over genes, such as antibiotic resistance. So, mutations and recombinations create cell diversity. Diversity is the raw material for evolution. Natural selection acts on populations to ensure that several organisms ensure that survival of organisms fit for a particular environment. So if you are resistant to every antibiotic in the world, but you're living in the wild, that might not be the best fit for you. But if you're resistant to every antibiotic in the world and you're living in a skilled nursing facility or a long-term care facility, that might be a better fit.